Aboard the NOAA ship Okeanos Explorer. We're coming live to you today from the middle of the Pacific Ocean. Right now we're about 350 nautical miles west of Oahu, and the ship is currently underway and steaming towards the Johnston Atoll unit of the Pacific Remote Islands Marine National Monument. And here we plan to conduct daily remotely operated vehicle dives and overnight mapping starting actually tomorrow. Tomorrow will be our first ROV dive through the end of the month. So today we have our science team leads here to tell you a little about what we have planned for this upcoming expedition and to take your questions and to answer your questions live on the air and tell you a little bit more about what we have planned. So we're going to start first with introductions. My name is Kelly Elliott. I am the expedition manager for NOAA's Office of Ocean Exploration and Research, and today I'll be serving as your facilitator, taking your questions and reading them to our onboard science leads so they can answer them live on the air. And here today I have with me.
I'm Chris Kelly. Hello, everybody. And I'm with the University of Hawaii. I'm one of the two onboard leads. I specialize in deep water biology, and I really enjoy trying to work on glass sponges.
And hi, this is Mashkur Malik. I'm one of the mapping leads on board, and I'm responsible for operating multi-beam systems and mapping, which basically provide us with the topographic and bathymetric mapping of the seafloor, which helps us to locate where we want to dive with the ROV.
And I'm Chris Ma. I'm from the Smithsonian's National Museum of Natural History in the Invertebrate Zoology Department, and I'm an expert on deep-sea sea stars, both their taxonomy and their evolution. So I know a lot about how they've evolved and all the many different kinds of sea stars there are.
All right, thank you. And we have our first question. And rather than going first to Chris for an overview, I'll ask the question because the question is actually being posed by our office, the NOAA Office of Ocean Exploration and Research. And the question is, why are you exploring the areas around Johnston Atoll, and what do you hope to find?
Would you like me to answer this?
Yes,
one?
please,
Okay.
Chris.
All right, so we did do 11 dives in the Johnston Atoll Monument, and the Johnston Atoll Monument is part of the Pacific Remote Islands Marine National Monument. It's one of the, it's a really amazing place, and it's about 750 miles southwest of Hawaii, and we want to explore this area a lot more than we were able to do in 2015 because there are so many seamounts, so many varied seamounts and structures down there that have never been explored before, so we want to give it another look.
All right, great. Thanks, Chris. We have a couple of other questions that have come in. The first one is from Anatoly Babkin, and he is asking whether any new capabilities were added to our remotely operated vehicle. We do have a two-body remotely operated vehicle that we explore with during operations. The primary vehicle is ROV Deep Discover, and then we also have the Sirius camera sled, and we do not have any new capabilities that have been added since our previous exploration activities. The primary capabilities of that vehicle are really powerful lights, high-definition imaging capabilities, and manipulators to collect specimens on the seafloor. All right, the next question that has come in. Okay, it comes from Shireen Gonzaga. This is going to go to our onboard biologists. Corals and other organisms in the deep ocean are adapted to high pressure from the water above. What happens to their shape and tissues when they are brought to the surface as specimen collections?
I believe that's a good question for Chris.
Okay, well, there's one thing, a lot of these animals don't have any air spaces in them, and pressure really has the most dramatic effect when there's an airspace. So, for example, certain fishes down in the deep sea actually have air-filled bladders that they use for buoyancy, and they can come up with some sort of severe effects that are referred to as barotrauma when they're brought to the surface. However, corals and sponges, they're all water and tissue, and really when you bring them up, they look pretty much the same as when you collected them down on the seafloor.
Okay, our next question has come in from Roberta Kirby, who says, on past expeditions, we've had a camera in the lab showing the processing of the samples. Could we do this again this time? It's very interesting to watch the process. I'm actually going to turn for a moment to one of our video and telepresence leads and find out whether we might be able to do that.
Yeah, we'll try to set something like that up, see if we can get that configured.
And did they hear that?
Yes, I am talking to the Facebook crowd.
Excellent. Thank you, Roland. Okay, well, while we are waiting for additional questions to come in, I did see one earlier. I'm not seeing the name listed, and it was a question about how middle school students can get involved in our exploration activities. And I think there's two primary ways. One is to go to the oceanexplorer.noaa.gov website. There's full coverage of this expedition online. There will be daily updates, topical weblogs, and there's already background essays. So you can go there to learn more about the science behind the expedition, as well as what's happening on a day-to-day basis. You can tune into the live video feeds. And I'll add that if you have a middle school science teacher who's particularly interested in what we're doing, our education team does actually conduct professional development workshops around the country. There's an education section of our website that you can go to sign up for one of those. All right, we do have more questions coming in. The next one is from Georgia Lopez, who asks, is it true that most of the ocean is still unexplored, even with all of the advances in technology that we have today?
Mashkor, you want to take one?
Sure. Thanks, Georgia, for that question. The ocean is explored or not explored is defined by what do we mean by that? So if we are just looking for a topographic map for the ocean, most of the ocean has been mapped at a very, very low resolution. But if you are trying to find the features which are of interest to the biology or geologist, only few percent, the estimate is less than 5%, has been actually mapped at a resolution where a geologist or biologist can look at the features of interest, for example, volcanoes or seamounts, to go down and explore. If we take it to the next level, where we are talking about how much of the seafloor has actually been imaged by an ROV or by an underwater camera, I think the percentages will be, in my estimate, will be even less than 1%. Comparing that now to the extraterrestrial and the other planets, they can use the radars, which unfortunately does not work for the Earth's oceans because radar waves cannot penetrate the sea. They have mapped the moon and Mars at a resolution far enough higher than what we can get from the Earth's ocean's seafloor right now. So that's basically answers the question, like, yes, the oceans are very, very unexplored, and only less than 5% of the seafloor has been mapped at a resolution useful for biologists and geologists.
And I would add that in terms of biodiversity, a great number of species remains to be discovered as well. So every time we survey an area like this, we see only a small percent, but every time we pull up a new coral or a new sea star, it's usually new or something that hasn't been seen in a very long time. So there's definitely a lot left to be explored in terms of animals and other organisms that live at the deep.
All right. Thank you, Mashkor and Chris. So the next question that we have is from Jane Skabortnik, asking what factors limit the dive duration of the remotely operated vehicle?
Do you want to take one?
Sure. That's an excellent question. The vehicle itself could certainly stay down for much longer than the daily daytime ROV dives that we conduct. However, we are limited largely by staffing on board the ship, and we choose to have the operating model where we conduct a dive every day. Our goal is to go to areas that have never been explored before or remain very poorly known, and to take that first look and get a sense of an initial habitat characterization. We then collect the data sets and make them publicly available as soon as possible to enable follow-on research and exploration activities. So we have a very specific goal that doesn't require us to stay down, for example, 2 days at a time. So it's the combination of the staffing on board the ship and the model that we choose to operate that limits the duration of our remotely operated vehicle dives. The next question that we have is from Connie Gildner, and it is pointed directly at Chris Ma. So, Chris Ma, do you think that you may find a new sea star on this trip?
Hi, Connie. It's good to hear from you from Facebook. So yes, every cruise that I've seen Okeanos engage in, we've seen evidence of some kind of new species of sea star. The last expedition, we saw that new cookie star, and there's evidence that there are other types of similar sea stars in the area. I look forward to bringing evidence back of several new species on this expedition. So looking forward to sharing all of that with you as the cruise progresses.
All right, thank you, Chris. We have a question that was posed earlier from Randy Huddlemeyer, and he has asked, do you have a wish list of creatures or any specific species that you hope to see on your expedition?
Well, I think both Chris Ma and I can both answer that question, Randy. Absolutely. I mentioned before that I'm very interested in glass sponges. And so there's a couple of species of glass sponges that I hope to see here. I don't know if we'll see them, but I'd be delighted if we do. Glass sponges have their name, or the name of a glass sponge is because their skeleton is made of silica, which can give them sort of a transparent type of structure. And they're very, very beautiful animals. They're very ancient animals that have been around for a long time. And there's a lot of glass sponges that are here in the Pacific and deep water that are brand new species and undescribed species. We are constantly finding new ones. So I've got a couple of some of these on my list, and we'll probably find some more that aren't on my list that surprise me. And Chris, what about you?
Well, certainly I'm looking forward to finding new species of sea stars, as I've mentioned earlier, but I think one of the great things about watching animals alive in their habitat is to watch them doing what they do normally. And so a lot of sea stars will find video of them feeding or engaging in some other kind of behavior, which we wouldn't ordinarily discover or observe from having collected them via nets or other means. So there's definitely a long list of things that, a wish list, as you say, of things that I really look forward or hope that we'll see, including maybe sea stars that feed on sponges. That'll be great.
So the other thing, too, is we're not going to disregard all the other cool animals. There's some fish that we certainly would like to see, some very, very unusual fish. For example, we've seen something called a sorceress eel only on 2 dives so far in the whole 3-year project. So I would love to see one more of these really peculiar eels. There are some cusk eels and some rat tails also that I'd like to see. And I will not forget all the corals and some of the other animals such as the arthropods. There are some amazing crabs and corals that are down here. And in fact, the last expedition, the specimens of one particular family of coral have already been submitted to the Smithsonian, and somebody's already taken a look at it. And this is in the family Chrysogorgiidae, and they've concluded that every single specimen of that family that we collected in the last 3 cruises are new species. So hopefully we'll see some more of those and get even more new species.
All right, great. We've got quite a few other questions that have come in. The first one, Chris Kelly, Michael Clark asked, do sponges count as being animals?
Yes, they do. They are animals, so they definitely count. They don't quite move around as much as some other animals, but they are most definitely in the Animalia, the kingdom Animalia. But they are a very ancient member of that kingdom, and there's also quite a fossil record of these sponges that goes back hundreds of millions of years.
Okay, the next question has come in from the NOAA Office of Ocean Exploration Research and asks, will you be exploring the water column in addition to the seafloor?
You want me to answer that? Well, I'll go ahead and throw it out. Yes, we will. We've got some water column dives that are planned on the second, well, let's put it this way, on the second dive that we're going to do here in the Johnston Monument. We're going to work in the water column at the end of the benthic or seafloor portion of that dive and spend a couple hours doing some transects in midwater. And hopefully we'll see some of the amazing animals, jellies and fish and other things that live in the midwater on already the second dive, and we're going to be doing this on several of the other dives during the cruise as well.
Thank you, Chris. And Mashkur, can you just briefly highlight how we regularly explore the water column as well with our sonars?
Yeah, so we on board the ship, we have the capability of imaging the water column using our sonars. It's not equivalent to observing the organisms, but on the aggregate, we can locate the aggregation of zooplankton and the aggregation of fish if we see them using our sonar system, and that can help position the ROV in the right location in the water column. And that water column exploration is happening all the time. Whenever we are out here, we turn our multi-beam system on. In fact, if you look at this screen, that's showing you the water column imaging that right now we are going over. And that provides us with the layers in the water. Yeah, so we will be exploring both acoustically and using imagery, using the ROV during this cruise, the water column.
All right, thank you, Mashkor. The next question has come in from Linda Stallnecker, who asks, do you think there will be some sea cucumbers in Johnston Atoll?
Yes, undoubtedly so. Thank you for that question. We actually we visited Johnston Atoll briefly in past expeditions, and we recorded the occurrence of at least one animal called Benthodites, and I'm certain that we'll see more in all of their fine sea cucumber glory, doing the things that they do. Possibly some swimming ones, possibly seeing some leaving their finely cleaned poop on the seafloor. So stay tuned for much sea cucumber goodness. I guarantee that.
Excellent. Thank you, Chris. All right. A question that was asked earlier by Hector Baron. He says he's presenting next week his master's thesis on sponge taxonomy in Gorringe Bank. Which glass sponges concretely are you searching for?
Right. So Hector, probably the ones that are of particular interest are in the family Euplectellidae to me personally, because that's such a diverse family. I've also done some work on an order that's called Amphidiscophora, and you probably know what I'm talking about, but I'm not sure what some of the other people know about that. But these are a couple of the groups that we're looking for, but there are so many unknown glass sponges down in deep water here in all of the major groups, both soft and hard sponges, that I'm sure we're going to see quite a few of them, and I'm sure we're going to see some brand new ones. So I'm just excited to see any of them.
Great. A question that came in earlier from Dave Tellman. He asks, are your containment facilities on the ROV sealed, or do the subjects just die when you bring them up? What happens to the subjects when we bring them to the surface?
Well, I guess I can answer that. The containment, well, the containers, if you will, have got a really good seal on them. So the water temperature remains quite cold when the ROV comes to the surface. But there, of course, is a very dramatic pressure effect and light and some of the other environmental factors. So while some of the animals actually come up and they're still alive. For example, we can see amphipods and some other crustaceans that are still moving around inside the boxes. But a lot of the other animals are not doing so well. And if you consider the pressure change that they have come through, they've come through a rather dramatic change that's more than the pressure experienced if you were in the inside of a fully charged scuba tank, 3,000 pounds per square inch. So they don't really respond to those changes very well.
All right. Thank you, Chris. The next question is from Lindy, I think it's Rauchenstein, and the question is, what kinds of physical measurements do you take as you dive, both from the remotely operated vehicle and from the ship?
So I guess I can take this question. Thanks, Lindy, for that question. The physical measurements, there are numerous sensors which are installed both on the ship hull and also on the ROV. And as we are going down, we are continuously using them to measure several of the parameters. And just to highlight a few of them, they include the water temperature, which is most interesting, most important one. Then we have depth and salinity, conductivity, sound speed, oxygen reduction potential, and also we have our turbidity measurement, which gives us a measure of how much suspended material is in the water column. So these are all these things that we are collecting as we are descending down using the water column, using the ROV through the water column. On the ship's hull, we also have these sensors, but they are not. They're only sampling the top surface layer of the water column. So that's why it's really interesting and important to have these measurements done by the ROV as it's descending down the water column.
All right, thank you, Mashkor. So the next question has come in from Yukiko Colahan. It's actually the second question. This is our operations officer's son, and he asks, what do the sea lilies feed on?
The sea lilies. So sea lilies are echinoderms. These, I believe you're talking about the stalked crinoids, which are echinoderms, and these have a sort of a cup with a lot of feeding tentacles around the mouth, suspended on a stalk. These are what's called filter feeders, and they present their tentacles up into the water, and they will capture organic food, small organic particles as the water currents bring them by. Their relatives, the feather stars, feed in the same way. So they basically, as far as I know, it's small organic particles and not, for example, small crustaceans or animals. But I suppose that we really don't know a lot about the feeding habits of many of these animals. Certainly some sea lilies, some of the more unusual ones, actually are carnivorous, but they're not very well known. So we'll see which ones we encounter, but most of them are feeding on small organic particles in the water.
Okay, thank you, Chris. So the next question has come in actually from Mimi Lobecker, and I'm going to do a shout out. Mimi is one of our physical scientists, and today happens to be her birthday. So happy birthday, Mimi. And she asks, what is Moshkor's favorite kind of seafloor terrain to map?
Thanks, Mimi, and happy birthday. My favorite terrain to map is flat, boring seafloor. And the reason is because I can see any issues with my multi-beam very clearly, and if there's any issues, I can figure it out. On a slopey terrain with a rough terrain where there's a lot of activity happening on seafloor, which is obviously very important for geologists and biologists, but my multi-beam sonar doesn't give me any clear indication what's happening and what's wrong with my multi-beam sonar. So my favorite terrain would be boring, flat seafloor.
Very interesting. Thank you, Moshkor. Okay, the next question is from Monica Sela, and the question is, why is water temperature one of the most important parameters to measure?
Yeah, so water temperature at the surface, and I'm going to use centigrade because that's usually what we work in. Water temperature at the surface out here is anywhere between about 25 to 30 degrees centigrade. When you get down to 2,000 to 3,000 meters, you're looking at between 1 to 2 degrees centigrade. And so you've got a whole transition sort of depth range where the temperature drops rather precipitously from the surface down to maybe about 1,000 meters, and then it kind of flattens out. Certainly some animals prefer a certain temperature range. Each animal is a little bit different. So basically some of the really deep animals do quite well in very, very cold temperatures. And some of the surface animals that are close to the surface, say 500, 300 meters, they can't tolerate the really cold temperatures. So in order for us to understand the whole picture of how the environment is, sort of stratifying communities down in the deep water, we need to measure temperature. And even though I talked about pressure a little while ago and that air is usually the thing that's affected most by pressure, one thing I did forget to mention is that even the very deep animals, even though they don't have air spaces, they have enzymes and metabolic processes that are affected by pressure. So when you move them out of their typical pressure range, their actual physiology can change dramatically, and that's probably why some of these animals die when they get to the surface.
All right, thank you, Chris. Glenda Salyers Chapman asks, what is the deepest depth you have probed and the strangest you have encountered?
So the deepest we've been with this particular ROV, and for me personally with any ROV, is close to 6,000 meters. This was in the Mariana Trench last year. And that is at the maximum depth limit of this particular ROV. And this ROV, it's one of the deeper ones. There aren't that many out there that can go below 6,000 meters. So that's the first part of the question. What was the second part?
The strangest animals you have encountered.
Strangest animals, boy. There's usually a strange member in every single animal group. I can't really go to one particular animal group. Maybe Chris, you've got a...
Actually, believe it or not, I think some of the strangest animal or organisms that we encounter at that depth aren't even animals. They're actually giant amoebas. There are organisms, protists called xenophyophorians and sort of related protists. These are single-celled animals. Single-celled organisms, like your household amoeba, the one that you find in freshwater ponds, but they are really big and they create large sedimentary structures that are actually macroscopic. And it turns out that there are studies that show that these amoeba actually capture animals. They can eat worms, they can eat all sorts of small sort of things living on the bottom. And they often create these very cryptic structures. Sometimes they're balls, sometimes they're sort of leaf-like things, but very strange giant amoebas living in the deep.
All right, thank you, Chris. So the next question we have is from Nora Lokshin, who asks, what can you tell us about the title of the expedition this year, the meaning of Laulima o Ka Moana? And Nora, thank you so much for asking this question. It is a very meaningful name. So Laulima literally means many hands and symbolizes cooperation, unity, and a shared vision. We use Laulima in the context of cooperation and partnerships, while Moana means the ocean. So without partnerships, the end goal of gaining more knowledge about the deep ocean would be difficult, if not impossible. So the meaning of this title is quite literally partnership for the ocean, and it highlights how important it is for us to come together and work on these initiatives together, as the efforts of many lead to the strength of the whole. So the next question that we have is from Benjamin Wilson. How in the world can we study behavior or even community ecology when we only see glimpses at these depths?
It's a good question. So I guess you got to start somewhere, and that's perhaps what we're doing. So we capture as much imagery as we possibly can of animals. The one issue, of course, with behavior, as you probably are aware of, is that when you illuminate an animal that's living down in 2,000 meters of water, for example, it's not used to any light whatsoever. So you have dramatically changed its environment, and the behavior that you're seeing also is undoubtedly affected by the light, at least those animals that have eyes. So whether or not you can get some idea of how the animal normally behaves, that's very tough to do. And actually, I don't have an answer for that. As far as just in general trying to understand the communities down there, again, we're getting a start. We annotate all the videos. We record all of the animals that are captured on video at each of these sites. We also record which animals are living on other animals, and we're trying to tease out as many relationships as we can.
I think it's important to note also that, remember that our mission mandate is exploration, not necessarily to answer one specific scientific hypothesis. So a lot of what we're doing is capturing incidents or occurrence of these behaviors so that scientists can come back later and put more detailed effort into studying them, so the communities and the behaviors. So oftentimes collecting the species and giving and figuring out that they're there is the first step to furthering our understanding. So we're all, as Chris said very clearly, it's the first step. And that's what we're here to do, is to explore and get all of that firsthand knowledge, and then let other people work out the rest with more data.
Thank you, Chris. Okay, the next question, and just note that we're approaching the final 2 minutes, comes from Chuck Thabal, who asked, do you see any anthropogenic debris at deep depths?
So Chuck, yes, we do. We saw some debris in the Mariana Trench. We've seen plenty of debris around the Hawaiian archipelago when we've done dives there, but not tons of it in the depths that we're typically diving in. So it's there, but it's not exactly all over the place. We still see plenty of pristine areas.
And we'll go for one final question. This one comes in from Valerie Drost, who asks, is there a relationship between the high pressure in these deep depths and the size of the animal species down there?
Okay, Chris, I think she was...
I've heard lots of different hypotheses about gigantism in deep sea animals. I don't know that I know of any that correlate pressure that might affect egg size or other factors. a lot of that is probably something that's very specific to different organisms. So I guess my answer is I don't know, but I don't think that's that there isn't a hypothesis. I'm just unaware of any that pertain to echinoderms, for example. So
Yeah, it's a very good question. The issue is that we just don't know at this point. So
it's also a fairly difficult way to measure influence, because pressure doesn't. I can't think of any immediate characteristic that correlates with pressure. So, so possibly yes, there could be some really interesting stuff there. So perhaps someone's PhD thesis someday.
Excellent suggestion. Well, thank you, everyone. Thank you for those of you who are tuning in online, who took the time to listen in and to provide some questions. We will be signing off tomorrow. Will be our first remotely operated vehicle dive. We'll be diving on Horizon Guyot. We plan to deploy the vehicle at about 8:30 a.m. Hawaii Standard Time, so please tune in then to learn more and to explore with us. And you can join us online by going to the oceanexplorer.noaa.gov website, and there will be a link both to the expedition website and the live feeds online. So please join us, and thank you so much for taking the time out.
yeah, thank you.
Thank you.
Thanks for your questions, screen grabbers.